Hi, everyone. Thank you so much for joining us. Before we kick this off, I do want to point out that we are taking audience questions. Please utilize the Q&A section at the bottom of your screen. Type in questions as they come up, and we will address those at the conclusion of today's presentation. So I'd now like to turn this over to Pam Genemere, our Client Success Manager here at Force, and she's going to give a brief introduction of Allarity Therapeutics. Thank you.
Hi, everybody. Thank you so much for joining Allarity Therapeutics webinar today. I want to say just thank you for taking the time out of your day to learn more about Allarity, the significant strides that they are doing, and about their lead drug, Stanaparib, and the impact that they've had on people's lives. The company was founded in 2004 and have made major strides with their lead drug. And I will turn it over to Thomas Jensen, CEO and co-founder, and Jeremy Graff, President and Chief Development Officer. They will take a deeper dive and lead the discussion as well as go through the presentation with you.
Yeah. Thanks, Pam. So welcome, everybody. I'm Thomas Jensen, and I will be, together with Jeremy, running through some of the most important slides and strides we have made in Allarity. We will be centering the discussion around our lead, our main asset, our novel ovarian cancer therapy called Stanaparib, and also our patient selection drug response predictor. We call this the gift of time. The first patient that was enrolled in our trial is now well past 14 months on treatment and still going strong. And when she entered the trial, she had maybe 2 to 4 months left, between 2 to 4 months left to live, according to the statistics. But since then, she's witnessed the birth of 2 grandchildren and experienced 2 Christmases. So that we think is amazing. We have 2 patients on drug for more than a year now, and we have seen amazing results that Jeremy will go more into detail with. So this is a legal statement. We're listed on NASDAQ, and I trust you're all speed readers. So we believe that we can have a very good future ahead of us with our novel drug. Stanaparib, it's a novel therapeutic in a very big market. We are, as I mentioned, seeing some amazing clinical benefit, and it's on top of that, it's a very well tolerated drug, an oral medicine that is given twice daily, just a tablet. So the patient can be at home and doing and taking the medication. No need to visit hospitals every day. So we also have instead of the other PARP inhibitors, it's a unique mechanism of action combining two pathways. And the patients treated are selected by our proprietary patient selection technology, TRP. So we will go into that also later. Our current market cap, as per our last Q, was around $5 million. And here you see comparison, the drugs, the space we're in, and the comparators ranging from smaller to very large pharma. So we think a very unique proposal here, and a unique drug with unique mechanism of action, and we're in a very large space here. So on myself, I'm a co-founder back in 2004. I got back into the company a year ago and a month, and together with Jeremy, we have been getting the company back in shape. We had several different classes of shares a year ago with various protection incorporated into them. We had negative shareholder equity and and also a bid price less than $1. So we had 2 NASDAQ deficiencies, no cash. But since then, we have now gotten in complete compliance with NASDAQ. We have raised almost $40 million and gotten rid of the almost all of the debt. So we are now ready to. We have a very good foundation to build on now, and Jeremy comes from the other side of the fence. He used to spend time in Eli Lilly, and then also later on in similar companies to us. So he brings a wealth of knowledge and helps us guide our strategy and clinical trials moving forward. We also have a very good board with expertise in each of their fields. So we are, I believe, guided towards a very good future. First, a few slides on our drug response predictor and what it actually does. As you see on the left here, cancer is very complex. This is a graph representing 680 non-redundant proteins, and this is the TNF alpha. When TNF alpha enters a cell in the textbook, 4 things happen and the cell dies. But if you actually look at what happens instead of what the simplistic version tells you, then it's a complex system of many interactions that there's no way we can fathom in our brains. But by using computing and good algorithm design, we can actually make sense of this complex knowledge and make it useful, which is our approach to this. So let's see what actually is in the data, not what we think is in the data, but let's look at what happens and let that guide us. So the tumor tells us what's important, and we have a few filtering algorithms along the way, and we end up with each DRP being somewhere between 50 to 400 different messenger RNAs that is upregulated or downregulated that we use as a score, and each patient gets a score between 0 and 100, depending on how likely you are to being a responder. The CDX here is relatively simple, and what other companies also were doing, we get a tumor from the patient, and we compare it to the gene expression to our DRP signature, and then we decide whether or not we will enroll the patient into the trial or not. So now Jeremy will take over and talk about stenopurib.
Thank you, Thomas. I just want to spend a few moments really describing what the drug does, how unique it is in the space, and really what our data are telling us now as we proceed through an ongoing phase 2 trial and head toward a registrational path we would like to finalize this year. Let's go to the next slide. So the drug stenopurib is unique in the PARP inhibitor class. It doesn't inhibit just PARP. It also inhibits an enzyme called tankerase, a related enzyme. Tankerase plays a role in the Wnt cancer cell survival pathway. So as a best-in-class orally administered small molecule inhibitor of both of these targets, we know that we can disable DNA repair via the action of the PARP inhibitory activity. That leads to cancer cell death. We also know that we can disable the Wnt pathway by blocking tankerase, which prevents cancer cells from using that crutch to stay alive in the face of all the DNA damage that we've induced with the therapy. We know in our ongoing trial, we have very heavily pretreated patients. Thomas mentioned earlier, one of our very first patients. These patients have all been treated with at least 3 lines of therapy before entering our trial. They were all selected using the stenopurib drug response predictor. And so we've ring-fenced already the patients that we think will get the greatest clinical benefit. Some of our patients have been treated with 6, 7, 8 prior lines of therapy. That's how heavily pretreated they are. And one might imagine at this point in the patient's journey, they have typically 2 to 4 months before they would progress through any therapy and perhaps end up in hospice. So to be able to see durable benefit in this patient population, one, is a high bar, and two, it's a high bar that we're crossing with a number of our patients. The development of stenopurib started in a large pharmaceutical company in Japan called Eisai, and in their hands, they had run a phase 1 trial. And it was within that phase 1 trial, they had first seen activity in ovarian cancer patients. They also saw activity in other cancer indications, including pancreatic cancers. So when we took over at Allarity, we were able to look at that phase 1 data set from Eisai and realize that our drug response predictor, specific for the drug stenopurib, could actually find the patients in a blinded retrospective study who had gotten clinical benefit. And really, that's what provided the spark for our enthusiasm to in-license the molecule. We now have global rights exclusively from Eisai to develop the molecule. We've secured the drug manufacturing in a GMP level way with Lonza. And we also have, with our DRP in place, we were just awarded in Europe the patent that allows us to treat patients with DRP and patients who are DRP positive with stenopurib, and that gives us a great IP position as well. Let's go to the next slide. So these are just a little bit of data from the ongoing phase 2 study, data that we've already pushed out into the public domain, but data that we want to highlight. So we treat, in this case, twice daily. We give patients capsules that they take in the morning and in the evening. These, again, very heavily pretreated patients who had already seen, in many cases, PARP inhibitors. And as it turns out, patients who are for the most part, platinum-resistant patients. So they're really out there and have very limited options afforded to them for continued treatment. What we've seen and what's depicted here in green is a patient that had a complete response and a confirmed complete response, meaning the disease, as we could measure it, went completely away. Then, when I say confirmed, at multiple scans thereafter, we also saw the disease was completely melted away. To have a complete response in this particular patient population after having already been treated with a first-generation PARP inhibitor really is unprecedented. A couple other highlights from the study. We have 6 patients that have been on therapy more than 16 weeks. 16 weeks in the clinical realm for these kinds of patients is a meaningful guideline, a meaningful milestone for these patients to reach. 4 have gone beyond 30 weeks, and we have continued with 2 patients that Thomas talked about before that are well beyond a year now. So you can tell just by the fact that these patients can take this drug twice daily for well more than a year that it's very well tolerated. The beauty of this drug is it also seems to, not only is it well tolerated by the patients in real time, but it doesn't seem to provide some of the salient toxicities that have afflicted the first generation PARP inhibitors, the myelotoxicity, the bone marrow toxicity, that a lot of these folks actually stopped taking the original PARP inhibitors because of. So we're excited about the way the drug looks. We're excited about it being very well tolerated. We're excited about the fact that it has a unique biochemical inhibitory activity, all of which gives us an opportunity to develop this drug and push it towards a registration path with the FDA. Let's go to the next slide, Thomas. So just 3 very quick patient vignettes. I'd like to show the actual experience and the kind of treatment history these patients have been through. This is our first patient. The patient has been on therapy more than a year. You can see in the bluish ribbon at the top, the patient is considered platinum resistant, had prior PARP treatment, has gotten the most recently approved drug in the ovarian cancer space, an antibody drug conjugate called Allarity. The patient was also treated with bevacizumab, the anti-angiogenic antibody, and the patient has a germline mutation in the DNA repair enzyme called BRCA1. These are the kinds of patients that typically and traditionally get on PARP inhibitors and often get benefit from PARP inhibitors. The graphic on the right-hand side, the green line shows you the patient's tumor burden. So you can see that the dotted line is their baseline tumor burden when they enter a trial, and you can see that the patient's tumor burden has shrunk during the time on treatment. You can see on the left-hand side how heavily pretreated the patient was. So surgery with then platinum and taxanes, that's pretty typical, followed by maintenance taxanes, then recurrence, treated again with platinum, but now also with a PARP inhibitor called olaparib, then treated with mirvetuximab, which is the other name for Elahere, carboplatin, bevacizumab. So this patient's very heavily pretreated. Might expect 3 to 4 months on a therapy at best, and yet we're out more than a year, and it's extremely well tolerated. Let's go to patient number 2 as a different example. The first patient I just showed you was a patient who would classically get a PARP inhibitor because they have a germline mutation in the DNA repair enzyme BRCA1. This patient, patient 2, does not. This patient would typically not get a PARP inhibitor because the patient has a normal BRCA gene, a wild type BRCA gene. So the patient came to us as PARP inhibitor naive because the genetics of the patient wouldn't have suggested they would get treated. This is an important point. This allows us to see activity in a patient population that otherwise doesn't really have any great options. Without the BRCA mutation, these kinds of patients typically will never see a PARP inhibitor. They would see an anti-angiogenic and maybe just other standard chemotherapies with all the attendant toxicity that those chemos provide. So again, very heavily pretreated. The patient was platinum resistant, but this patient tells us a different story than what PARP inhibitors traditionally can tell us, because we see this clinical benefit more than a year, even though they have a wild type BRCA gene. Let's go to the third example. This is our confirmed complete responder. So this patient came to us again, very heavily pretreated. You can see the treatment history here, starting all the way back to 2015. The patient was considered platinum resistant. The patient had previously been exposed to a PARP inhibitor as well as a checkpoint inhibitor. The patient has a mutation in the BRCA2 gene, so an affiliated DNA repair gene. The patient very quickly on our therapy went to a complete zero in terms of the disease burden and maintained that for well more than 30 weeks. This is an unprecedented clinical event in patients who are PARP retreated. Next slide. So just as a quick summary of what we have shown you today, and to help understand the enthusiasm we really have a molecule here that I think is very distinct from the other PARP inhibitors. The PARP inhibitors as a class are already out there in a $9 billion plus market that's only ascending. Ours is very different. Ours has a favorable and very unique safety profile. As I said before, we don't have the bone marrow tox that afflicts a lot of the first generation PARP inhibitors. We also have efficacy in patients that historically you would never see efficacy in with a PARP inhibitor. And that efficacy is durable. That's the most important part. It's durable. We give people the opportunity to stay on an easy to take drug for a long period of time. So we're excited about that unique aspects of the molecule, but we're also excited about what we're putting in place to execute on this molecule. The molecule, as so many are, has been in clinical development for a while, but looking for the appropriate signals. We think we have those signals with this ongoing phase 2 study, and that gives us the opportunity to construct and finalize a registrational path for this molecule and to accelerate into that path starting sometime this year. And we expect as we carry through this year and into 26 and through 26, there will be multiple data readouts. In other words, multiple inflection points of value that we can accumulate as we drive this molecule more and more closely to FDA approval. So we think that really represents a really huge opportunity in the advanced ovarian cancer space, where really there's very limited opportunities for therapeutic intervention, and certainly limited opportunities for those that are so well tolerated. Next slide. I think that now gives us the opportunity to take questions, Callie.
Yes. So let's, can you talk about what your mechanism of action is?
Thomas, you want me to handle that, I presume?
Sure.
All right, fair enough. So the PARP inhibitors as a class inhibit DNA repair. When we damage DNA repair mechanisms in cancer cells, especially cancer cells where affiliated symbiotic repair mechanisms are already disabled, in other words, in people that have a BRCA defect, we can create cancer cell death. One of the things, though, that we see in the field, and we see this in a lot of cancers, but in ovarian cancer specifically, some of these patients, their cancer cells learn how to survive even in the face of inhibition of PARP. Very often that involves activation of the Wnt pathway. Well, as it turns out, as we learn more and more about our molecule, we now recognize that our molecule also inhibits that Wnt pathway that is enabling progression of these ovarian cancers beyond the first stage, if you will. So our mechanism is unique because we're hitting the first target to disable DNA repair, and then we're hitting an affiliated target and pathway that are actually driving progression through those first generation PARP inhibitors. And it's actually a pathway that's activated in many, many, many advanced cancers. So we can contemplate how this gives us an opportunity to imagine going beyond just ovarian cancer. This pathway is activated in upwards of 80% of colorectal cancers, for instance. So very unique mechanism of action hitting both enzymes and both targets. It allows us to really consider different and novel ways to use this molecule. And one of those was exemplified by a couple of the examples I gave you. Normally, a patient with a BRCA wild type gene would never get a PARP inhibitor. In our case, we have a patient, one of our very best patients, who was BRCA wild type, who was out on the therapy more than a year. And that tells you that that other piece of our mechanism, the tankerase or Wnt pathway inhibitory action, is really operative in those patients.
Thank you. And what have your conversations been regarding your endpoint with the FDA?
So the endpoints in this space typically are going to, in early to mid clinical work, be related to progression-free survival. So the time it takes from when a patient joins our trial to the time when maybe their disease progresses. We would like to see that be eternal, infinite. But the reality is these are very heavily pretreated patients. They have advanced disease. Typically their window of time to be on a therapy when they're this heavily pretreated is only in the order of a couple of months. So what we want to be able to do, and the endpoints we'll play to as we carry forward, are to extend that interval between when they get on our therapy. If they should normally naturally come off 2-3 months in based upon what they would otherwise get, and we're out 6, 8, 10, 12 months, like our two best patients are now, out beyond 12 months. That's a great signal that we're going to drive an overall impact on overall survival, which is obviously what we're all mostly interested in. But progression-free survival is a bit of a surrogate marker for overall survival in this space, and one more often than not is used as the primary endpoint when you're reaching for registration.
And do you expect your phase 3 to be patients in severe shape, or might you treat a less severe patient population?
I think it's possible that you could imagine both. You actually think about the unique mechanism of action of the molecule and its safety profile. We have the opportunity to be in very advanced platinum resistant, possibly even platinum refractory patients who have absolutely no treatment options available to them. We also could imagine being pulled forward in the patient's therapeutic journey, maybe even supplanting some of the first generation PARP inhibitors and those that reside in and drive the $9 billion market we mentioned before. So there's lots of opportunity for this molecule. Typically, in the cancer space, the most expeditious path to registration is in more advanced patients. It's just the trials proceed more quickly. You get answers more quickly. But to go earlier in a patient's journey is certainly a high value opportunity from an investment standpoint. It just takes a little longer in that space to drive a registrational path.
And what have your conversations with large pharma been? Do you expect to take this conversation all the way through phase 3?
I think you have to, as a company, expect to drive this yourself. I think that you have to set yourself up to drive it all the way. You can never predict what somebody else is going to want to do. But our game plan is ultimately to drive enough interest based upon the unique activities and benefit that this molecule provide, that the big pharmas will come to us looking to partner. They're going to want a game-changing PARP inhibitor. There are second and third generation PARP inhibitors being explored right now by big pharma. We have a safety profile that we think very comparable to those, but we have a unique mechanism that gives us a different stance and different opportunities than those molecules that are currently in development within big pharma provide. So we will absolutely set ourselves up to take this all the way forward, but we will, I'm sure Thomas would agree with this, be very opportunistic should a strategic partner come along and want to really help accelerate this.
Can this therapy also be applied for colon and liver cancer?
We think it could. So especially with the fact that it's a Wnt pathway inhibitor in addition to a PARP inhibitor. The Wnt pathway is so commonly involved in a lot of very advanced cancers. And like I said before, genetically, we can estimate that up to 80% or so colorectal cancers actually activate this pathway. Liver, clearly there, pancreas, prostate. There's just an inordinate, there's really quite a wide berth of opportunity based upon that recollection. I've been in the pharma world, big pharma for almost 2 decades, and then biotech for another decade thereafter. I can tell you we've always wanted a nice Wnt pathway inhibitor that could be well tolerated. The industry hasn't had one. I think we have one.
And what about orphan drug or compassionate use designations?
So those are two very different things. Compassionate use, I think Thomas and I and the leadership of Allarity would always consider the possibility of compassionate use. Look, we're in this game to try to see if we can help patients out. If there's an opportunity to do that, I think we want to consider it. And I will tell you from a personal standpoint, being in a smaller company where decision-making is more agile allows for that to happen maybe more readily than you would see in a big pharma company. So that's one thing. I think if we're thinking about orphan drug, I think I would expand that to just any regulatory path that is advantaged. An orphan drug path certainly would be advantaged. There are other accelerated paths to approval that allow us to link up and have more robust, quicker, more frequent conversations with the FDA. We're looking at all of those opportunities. Of course, our clinical data opens those opportunities, but we have to look at all of those because if we can capture an advantaged path to registration and a lot more insight and productive relationships with the FDA, we're certainly in favor of that.
And do you expect off-label use?
I think the reality of the world is off-label use happens. We can never position ourselves to look for that or to promote that, right? But anytime docs get an opportunity to understand how a drug works, they can think creatively on their own about how it might be applied in different situations.
And let's a few business questions here. So where do you fit in regards to valuation compared to your peers?
Should we take that one?
Yeah. We see that the companies we compare to, we are heavily undervalued compared to the companies that have unique drugs or differentiating technologies like our DRP.
And do you think running a company that focuses primarily on women has been detrimental to the valuation?
Honestly, no, I don't think so. What has been detrimental is the mishaps of happening previously with entering into some suboptimal financings and also the refusal to file we got on a tanked program, a program we no longer have. I think that has been the major reason that the market lost trust in previous management.
There are a couple questions about that. Have you since corrected what the old management team has done? And can you also talk about your capital structure? You know, has that been cleaned up from the old management?
Yeah, we can say that a year ago we had no funds and we looked into being delisted and or going bankrupt, honestly. We had common shares, we had Series A preferred shares, and we had different warrant structures also. The Series A preferred shares had downward protection. So anytime shares were issued at a lower price, they would get a new batch of shares at a lower price that matched the lowest price. So no matter what happened, they got more and more shares that they could just sell out, and that created a death spiral, proving it very difficult to find a good investor base for that. So that was early last year. We're now in a situation where we have only common outstanding, only common shares. We have only very limited debt left in the company. We have secured one way into 2026, and our shareholder base is primarily now retail.
And how have you been able to raise so much money?
So we have been because we have been in shelf registration. So we've had an ATM. We have been using. We got out of baby shelf limitations, so that has helped us tremendously.
And when you founded the company, were you data or AI focused or more of a traditional medical research?
So when we founded the company, I founded the company with my former professor. This was back when in 2004, when machine learning was big. We are not using machine learning per se, but a part of it was figuring out in the lab, which was my area. How do we extract messenger RNA from a cell and do it full length, and how do we amplify this messenger RNA in a way so we can actually start building tools around it and measurements so that we can have linearity in what we see and look at the different dynamics of the gene expression inside the cell. And the other part was how do we, what happened was that we saw in cell lines, NCI 60 National Cancer Institute had cell line panels running, where all researchers or drug companies could send out their drug to be run by these cell lines, which was from actual cancers grown in lab flasks. And we were able to see that there was a robustness in this signature that made us able to predict outcomes just by looking at biopsies and the cell line data. So a part of it was bioinformatics, and the other part was molecular biology intertwined to build this.
Well, that concludes the questions. Before we sign off, I just want to turn it back over to you, Thomas and Jeremy, for any final words for the audience.
I just want to say we're always happy to do a follow-up and discuss this in further detail once you've digested what we have been discussing here. So always welcome and always happy to discuss further.
Great. Well, thank you everyone for joining. And please reach out to me. Everyone out there has my email address. If you want to learn more, be introduced to the team, I'm happy to facilitate that. And I hope everyone has a great day.